Hello everyone, my name is Josué David Hernandez Varela. I'm a current student in the PhD of Nanoscience and Micro Nanotechnologies of the Instituto Politécnico Nacional in Mexico City, Mexico. In this opportunity, I have the pleasure to present my poster titled Crystallographic Study of Cellulose Nanoparticles from Garlic and Agave through High-Resolution Electron Transmission Microscopy, or Hr-Tem. The crystallography study of a sample is very important when we can found different characteristics of the sample, means of the molecular composition or the physical interactions in a sample. For example, different techniques can be used: X-ray diffraction, spectroscopy, infrared spectroscopy, or Raman spectroscopy. For this case in particular, we choose to use high-resolution electron microscopy, or Hr-Tem, to elucidate the structure of a crystalline material. In our case, we use biological materials extracted from different sources. For example, in this case, agave and garlic. The agave and garlic samples have different compositions in the cell wall: lignin, hemicellulose, and cellulose, as we can see in the figure 1. But the only part of this composition that is crystalline is cellulose. To achieve the objective of this study, we use a very simple but complex methodology to obtain the cellulose nanoparticles from agave and from garlic. As you can see in figure 3, we show the experimental design for the obtention of the cellulose nanoparticles. In the first place, we use a pretreatment. After that, isolation part. And finally, we use a ball milling technique to achieve particle size less than 100 nanometers. In the case of the simulation and also the calculation from the micrograph, we use 3 different software of image analysis. Following the methodology that we show in the experimental part, we obtain the following results. In the first case, we can see the image in the right side of the screen. As you can see, they are the image from the cellulose nanoparticles from garlic and from agave. In the first part, we can see that the nanoparticles have a characteristic hydrophobicity that is well known, and that provokes a reagglomeration of the samples when are deposited in the 10 grid. In the second row of the image, we can see that garlic nanoparticles present more rugosity and also small sizes in comparison with the cellulose nanoparticles from agave. In the third row, we can see that the cellulose nanoparticles have characteristic crystalline regions that are common in this kind of materials when they are obtained from an isolation. And we can see that these particular crystalline regions have a very well-defined Fourier transform. With the inverse of the fast Fourier transform, we can create a more specific pattern in which the cellulose chains are more clean, and we can make very easily the calculations. Finally, we can obtain from the simulation that the distances are very similar between them, but the angles are the main part in the simulation and are characteristic from the triclinic or the monoclinic structure. Founded that cellulose from agave is more monoclinic and cellulose from garlic is more triclinic. With this important investigation, we can achieve the same results using spectroscopic techniques, but only using one image from a bunch of images that we can obtain in the TEM microscope. Thank you very much, all the participants in this symposium, and also for the Institute for allowing me to present part of my investigation. Thank you so much in advance, and see you soon.